If you look at human physiology, especially when we talk about reproductive biology, we're kind of conditioned to look for the rhythm. We're always looking for the cycle.
Right. The monthly clock, the phases, the ebbs and flows.
Exactly. We talk about fluctuations, windows of opportunity, the rise and fall of hormones. And the female system is, it's a masterclass in timing. It is fundamentally beautifully cyclical by design.
It absolutely is.
But today, we're going to flip that logic on its head. We're looking at a system that is engineered for the exact opposite of a cycle. We're looking at a system built for relentless, unwavering constancy.
It's a complete paradigm shift, really. Today, we're doing a deep dive into the male reproductive system. And if the female system is about timing, which it is, the male system is all about readiness.
Persistence.
Persistence is the word. It's this incredible engineering marvel designed to maintain a continuous production line of millions of cells, not once a month, every single day, for decades.
And it has to do this while balancing these incredibly potent hormonal signals that, they affect everything from muscle mass to behavior to metabolic health. It's not just a local system.
No, not at all. It's systemic. And the wild thing, and this is what really struck me going through the sources today, is that when you look at the nuts and bolts, you realize this constancy isn't the default setting. It's not the easy option.
It's an achievement.
It is a physiological high-wire act. It takes a huge amount of energy and regulation to maintain that steady state.
So we got a massive stack of notes here. We're synthesizing the mechanistic details from two absolute Bibles of the field. We're looking at Ganong's review of medical physiology, specifically the chapter on the function of the male reproductive system, and we're blending it with the systems-level perspective of Guyton and Hall from their chapter Reproductive and Hormonal Functions of the Male.
Two absolute heavyweights. And I think it's important to say right up front, what we're doing here is an interpretation and a synthesis. We're building a conceptual map for you.
Right. This is not a substitute for opening the book.
Not at all. In fact, our goal with this deep dive is to help you read those chapters better. We want to give you the scaffolding, the framework, so that when you open Ganong or Guyton, you see the structure, you see the why behind the what, and the details they stick better.
Yeah, we want you to go back to the source material with fresh eyes, with a kind of mental model already in place.
Exactly.
So the mission for this deep dive is to answer one central question. How on earth does the body pull this off? How do you maintain a factory that never shuts down, keeps its product at a temperature different from the rest of the body, and this is the part that blows my mind, protects that product from its own immune system?
That immunological piece is going to surprise a lot of people. It's one of those concepts where once you really get it, you never look at the system the same way again. It changes everything.
Okay, so here's our roadmap. We're going to start on the factory floor, the actual structure and the environment, temperature control, barriers, that kind of thing.
The architecture.
The architecture, exactly. Then we'll look at the product itself, spermatogenesis and the 74-day production cycle. Then we'll go up to the control room. We'll look at the endocrine loops.
And the management layer, the hypothalamic pituitary axis.
Right. Then we'll get into the molecule of man testosterone and the really crucial distinction between it and its more potent metabolites. And finally, we'll wrap up with the mechanics of delivery and some fascinating clinical correlates that really bring it all home.
Sounds like a plan. Let's get into it.
Okay, let's unpack this. Let's start with the architecture of continuity. When I read the description of the testes in Guyton, I was honestly just struck by the sheer scale of the plumbing. You look at a diagram and you think, okay, it's a solid organ, maybe the size of a large walnut.
It's completely deceptive. From the outside, the testes looks like a solid, encapsulated organ. But if you were to, look at the microscopic structure, it's not solid at all. It's a tangle. It's a mass of coiled tubes.
And Guyton says up to 900 coiled seminiferous tubules.
900. And that number matters. But what matters even more is the length of each one. If you unraveled those tubules, each one is about half a meter long.
Wait, okay, so do the math on that. That's roughly 450 meters, almost half a kilometer of tubing crammed into that tiny volume.
Exactly. You have this absolutely immense surface area packed into a very, very small volume. And that surface area is the factory floor. That's where the germinal epithelium sits.
So that's the whole point of the coiling, just to maximize real estate.
It's all about real estate. If you didn't have that coiled structure, you simply couldn't produce the sheer volume of sperm required for human fertility. We're talking about producing, on average, millions of sperm per day. You need a lot of space for that kind of production.
And the plumbing doesn't even end there. After the factory, there's a whole, shipping and handling department? The epididymis?
which, again, is just another tube, but an incredibly long one.
A 6-meter long tube, is that right?
It is. 6 meters, or about 20 feet. It's coiled so tightly you'd never guess. And this is a crucial distinction that Ganong makes, which is often missed in, a quick overview. When sperm leave the testes, they aren't actually ready to go.
So they're not functional yet.
They're morphologically complete, they look like sperm, they have the heads and the tails, but they are functionally immature, they can't swim properly, and they can't fertilize an egg.
They're duds at that point.
Effectively, yes. They have to travel through this 6-meter finishing school of the epididymis. This transit can take anywhere from 18 to 24 hours, sometimes even a few days, depending on the flow rate.
And what's happening during that commute?
The chemical environment of the epididymis is changing the sperm membrane. It's adding and removing proteins, altering the lipid composition. It's basically giving them the potential for motility. It's installing the software, so to speak.
Okay, potential is the key word there. You're emphasizing that.
It is, because even though they learn how to swim in the epididymis, the environment there actively suppresses them. It inhibits them.
So wait, why teach them to swim and then immediately tell them to stop?
It's all about energy conservation. Think about the scale of a single sperm cell. It's tiny. It has a very, very limited fuel tank, a finite amount of ATP in those mitochondria.
Right.
You don't want them revving their engines and burning out all their fuel before the race has even started. So the epididymis is this brilliant paradox. It grants the ability to move, but then it chemically forbids that movement until the moment of ejaculation. It keeps them in a state of suspended animation.
That is just incredibly smart design. It's like keeping a fleet of cars fueled up and ready to go, but with the parking brakes locked on until you get the green light.
That's a perfect analogy. The parking brake stays on until they're mixed with the fluids from the prostate and seminal vesicles.
Okay. Now, before we get any deeper into the cells themselves, we have to talk about the working conditions in this factory, because there's a very specific environmental requirement that seems, almost counterintuitive to mammalian biology.
You're talking about the temperature trade-off.
I am. The body's core is 37 degrees Celsius. We fight tooth and nail to keep it there. We shiver, we sweat, we have all these mechanisms just to maintain 37 degrees. But Ganong points out that spermatogenesis, the actual making of the sperm, just crashes and burns at that temperature.
It requires about 32 degrees Celsius to work properly.
So that's a 5-degree differential. In physiological terms, that's a canyon.
It's huge. Physiologically, that's a massive gap. Most enzymes in the human body are finely tuned, optimized for 37. But the specific enzymes involved in sperm production are optimized for 32. So the male body has to solve a basic thermodynamics problem.
How do you keep one organ 5 degrees cooler than the furnace it's attached to?
Exactly. And the first, most obvious solution is location, hence the scrotum.
Right, you hang the production facility outside the main building where it can get some air.
It allows for air circulation, evaporative cooling, and even muscle action. The cremaster muscle can pull the testes closer to the body for warmth or let them hang lower to cool off, but there's a deeper, much more elegant mechanism at play here that I found fascinating in the reading.
This isn't just about hanging them outside.
No, it's about the plumbing, specifically the blood vessels. This is the countercurrent mechanism.
Okay, this is pure engineering. I've heard this principle applied in other systems.
It is. It's the same principle used in industrial heat exchangers, in the gills of fish, and even in the legs of Arctic birds to keep their feet from freezing on the ice.
So walk us through this, because I think most people just assume blood flows in and out like any other organ.
So you have the spermatic artery coming down from the abdomen to supply the testes with oxygenated blood. That blood is coming from the body's core. It's hot. It's 37 degrees.
Right.
If that hot blood hit the delicate machinery of the seminiferous tubules directly, it would cook the factory. Sterility would be almost immediate. The germ cells would die.
Okay, so that's the problem. What's the solution?
The solution is that running right next to that artery, not just next to it, but literally wrapping around it in a complex mesh, is a network of veins called the pampiniform plexus.
And these veins are bringing blood back up from the testes.
Exactly, and because that blood is coming from the testos, which has been hanging outside in the cooler scrotum.
That venous blood is cool.
It's cool. So you have hot 37-degree blood flowing down in the artery and cool, say, 32-degree blood flowing up in the veins, separated only by a very thin vessel wall. Heat naturally follows the gradient. It moves from the hot artery into the cool vein.
So the heat is transferred across before it even reaches the organ itself?
Precisely. It's a pre-cooling system. It's a heat exchanger. By the time the arterial blood actually gets to the testicular tissue, it has already offloaded most of its excess heat to the venous blood returning to the body. It arrives pre-cooled to the perfect 32-degree working temperature.
This is brilliant. It effectively creates a thermal bypass around the body's core setting.
It does, and it's efficient too. The venous blood, as it returns to the body, gets warmed up in the process, so you don't shock the core with a slug of cold blood. It preserves core heat while simultaneously protecting the testes. It's a physiological win-win.
But this also highlights just how fragile this whole setup is. It relies on this delicate balance. Ganong mentions that if you disrupt this, say with cryptorchidism, where the testes don't descend and they stay up in the warm abdomen, the tubular walls just degenerate.
You get irreversible damage. The cells that are supposed to make sperm, the germ cells, they just undergo apoptosis and die off. Interestingly, the Leydig cells, the ones that make testosterone, are a bit hardier. They can often survive the heat.
So you could have a situation where a person masculinizes normally at puberty because they have testosterone, but they're completely sterile because the germ cells are gone.
Exactly. It's a classic clinical presentation. Normal secondary sexual characteristics, but azoospermia, meaning no sperm in the ejaculate.
Which brings up the lifestyle factors. This isn't just about anatomy, it's about behavior. You read about hot baths, laptops, tight clothing.
And Ganong actually cites data on this. Hot baths, we're talking 43 to 45 degrees Celsius for 30 minutes a day, can cause sperm counts to plummet by as much as 90%.
90%. That's a massive drop.
It's huge. Now, I have to be very clear here. You should not use this as a form of birth control.
Less. Yeah. Honey, I took a bath, we're good.
No, absolutely not. It is nowhere near reliable enough to be contraception. There are still millions of sperm left, even with a 90% drop, but it is certainly enough to cause subfertility if a couple is trying to conceive.
And even just tight clothing that holds the scrotum against the warm body can raise the temperature enough to negatively affect counts. It just shows that even a few degrees really, really matter.
They do, and there was that weird little factoid in Ganong about seasons that I didn't expect.
The winter effect.
Yeah, the winter effect. It turns out that sperm counts in men in temperate climates are often measurably higher in the winter than they are in the summer.
And this holds true regardless of what they're wearing.
That's what the data suggests. It implies there might be some kind of ambient environmental tuning going on, perhaps mediated by the pineal gland and light cycles, which we can touch on later. But it also just reinforces this core principle. This system is incredibly sensitive to thermal conditions.
Okay, so we've got the factory built and the temperature controlled, but there's another layer of security, a literal barrier. The sources make a huge deal about the blood testis barrier.
This is arguably one of the most critical and underappreciated concepts in male reproductive immunology. It's fundamental.
Because usually in physiology, we want blood to flow freely to tissues. We want to deliver oxygen and nutrients and clear waste. But here, we're actively walling things off. Why?
It comes down to a question of identity. Think about it. Every single somatic cell in your body, your heart cells, your liver cells, your skin cells, they all have your full genetic code, your 46 chromosomes. Your immune system patrols the body and recognizes that code as self. It has been trained since birth to protect it.
Right. Anything that doesn't match that code gets attacked.
Precisely. But sperm are different. Through the process of meiosis, they have shuffled the genetic deck. They are haploid. They only have 23 chromosomes instead of 46. They express new, unique proteins on their surface that don't appear anywhere else in the body.
So to the immune system.
They look like aliens. They are genetically distinct from the man who produces them. If your immune system, your T cells and antibodies, were to see a developing sperm cell, it would recognize it as a foreign invader.
And it would attack it.
It would launch a full-scale assault. You would develop anti-sperm antibodies, and the immune system would try to destroy the germ cells. It would be a catastrophic autoimmune reaction.
So the body has to build a fortress.
It has to build a fortress, and that fortress is constructed by the Sertoli cells. These are the large nurse cells that line the tubules. They form incredibly tight junctions with their neighbors near the base of the tubule. They effectively zip the tissue shut,
creating a kind of VIP section.
We call it the luminal compartment. Everything inside that barrier where the sperm are developing is immunologically privileged. It's hidden from the blood, hidden from the white blood cells, hidden from the rest of the body.
And Ganong mentions this creates a unique fluid environment too. It's not just about keeping the bad stuff out. It's about concentrating the good stuff in.
Right. Because the blood is walled off, the fluid inside the tubules can be custom made. It's very different from plasma or interstitial fluid. It's low in protein, low in glucose, but it is incredibly rich in androgens, estrogens, potassium, and certain amino acids. You're creating a custom nutrient bath for these developing cells that the rest of the body can't touch.
It's amazing to think that the body has to hide its own reproductive future from its own defense system. It's like an internal witness protection program.
It's the fundamental trade-off of sexual reproduction. If you
Right.
want to create genetic diversity through meiosis, you have to create cells that are genetically unique, and if they're unique, they're foreign. It's a profound biological dilemma with a very elegant solution.
All right, let's move into the factory itself. Section 2, the production line. We've built the factory, we've cooled it down, we've secured the perimeter. Now let's actually make the product. Spermatogenesis.
The main event.
The first thing that jumped out at me from both sources is the timeline. I think there's a common misconception that because men produce millions of sperm a day, it must be an instant process. Like, you eat a steak and boom, more sperm are made.
Not at all. It's a long, carefully orchestrated process. The entire cycle, from a single stem cell called a spermatogonium to a fully formed but still immature sperm, takes approximately 74 days.
74 days. That's nearly 2.5 months.
Exactly. And this is vital for systems thinking and for clinical diagnosis. If a man gets a high fever today or is exposed to a toxic chemical or takes a new medication that impacts cell division, he might not see the effect on his sperm count until 2.5 months from now.
There's a lag.
There is a significant lag. You are always looking at a historical record of that man's health from the previous quarter. When we do a semen analysis in the lab, we're effectively getting a snapshot of what was happening to that man's body 74 days ago.
That is such a key clinical takeaway. You have to ask about history, not just what happened yesterday.
You absolutely do. Have you been sick in the last 3 months? Is a critical question.
Let's talk about the cellular cast involved here.
Right.
We've mentioned the Sertoli cells, the nurse cells, but they're also the Leydig cells. I'll admit, I used to always get these mixed up. What's a good way to distinguish them?
Okay, here's a good metaphor. Think of the seminiferous tubule as a house. The Sertoli cells are the mother cells inside the house. They are physically in contact with the developing sperm. They nurture them, they feed them, they protect them behind that barrier, they dispose of their waste. They are structural and supportive.
Okay, Sertoli inside, nurturing.
The Leydig cells are the loud neighbors outside the house.
Loud neighbors.
They sit in the interstitial space, the tissue between the tubules, and they just shout out testosterone. They are the chemical engineers pumping out the key hormone that keeps the whole neighborhood running. They don't touch the sperm directly, they just provide the hormonal signal.
Okay, Sertoli inside, nurturing, Leydig outside, shouting testosterone. That really helps.
Precisely. And they communicate with each other through chemical signals, which we'll get to in the endocrine section. But their locations and primary roles are distinct.
Now, regarding the actual sperm development, the journey from spermatogonia to spermatozoa, Ganong mentions a fascinating detail called cytoplasmic bridges.
Yeah.
What is that all about?
Yes, this is one of those beautiful, subtle details. As the germ cells divide, 1 becomes 2, 2 become 4, all the way up. They don't actually separate completely.
Usually in cell division, cytokinesis pinches the two cells apart and they go their separate ways.
Right, that's the final step. But here, the separation is incomplete. They remain connected by these little bridges of cytoplasm.
So they're literally holding hands throughout their development.
In a way, yes. It means that a whole clone of cells, all the descendants of that one original stem cell, develops as a synchronized team, not as a collection of individuals. They can share nutrients, they can share signaling molecules through these bridges. It ensures that the entire batch matures at the same rate.
So you don't have some cells lagging behind while others race ahead.
Exactly. It creates a cohort that moves through the stages of development in lockstep, and they don't disconnect from each other until the very final stage when they are released into the lumen of the tubule. It's a team effort right up until the moment of launch.
And the final product of that effort, the sperm itself. I mean, it is the absolute classic example of form follows function in biology.
It is a stripped-down, high-performance delivery vehicle. It has shed almost all of its cytoplasm and organelles. It's barely even a cell anymore in the traditional sense. It's a payload with an engine.
So you have the head.
which is really just the payload. It's tightly condensed DNA, the genetic package, and it's wearing a helmet, a specialized cap called the acrosome.
The acrosome. That's basically a modified lysosome, right?
Essentially, yes. It's a membrane-bound sac that is jam-packed with powerful enzymes, hyaluronidase, and various proteolytic enzymes. This is the drill bit. When the sperm reaches the egg, it has to get through the egg's protective outer layers. The acrosome releases these enzymes to design a path and allow the sperm to tunnel through.
So without that acrosome, the DNA never gets in.
Never. The sperm would just bounce off.
And then, of course, the tail.
The engine.
The flagellum.
Yeah.
It's this beautiful molecular machine. It's powered by a sheath of mitochondria that are wrapped tightly around the base of the tail like a helical coil. They just pump out ATP to fuel the movement.
And the structure of the tail itself is that classic axoneme, right?
The central skeleton. Guyton describes it as 11 microtubules arranged in that specific 9+2 pattern that allows for the sliding, whipping motion that propels the sperm forward. It's a marvel of cellular engineering.
Ganong threw in a really specific molecular detail here that caught my eye, a molecule called JE.
Ah, yes, germinal angiotensin converting enzyme.
Which sounds a lot like the ACE we talk about for blood pressure medication.
It's in the same family. We usually think of ACE in the lungs and kidneys, converting angiotensin to angiotensin II to regulate blood pressure. But it turns out sperm have their own special version. And if you create a knockout mouse that's missing this specific sperm ACE, the mice are completely sterile. It just shows how nature reuses and repurposes tools. Oh, this enzyme works for blood pressure.
Yeah.
Let's tweak it and use it for fertility too.
Evolution is a tinkerer, not an inventor. It grabs whatever tool is handy on the workbench and adapts it for a new job.
Okay, so the sperm are built, they get released from their cytoplasmic bridges, they leave the testes, but as you said before, they still can't really swim, they're not ready. They travel the epididymis, get their final software upgrades, and then they wait for the signal.
Correct. They're stored primarily in the tail of the epididymis, in that state of suspended animation, waiting for the call to action.
All right, let's move upstairs from the factory floor. Section 3, the control room. Endocrine regulation. This is where the brain starts talking to the testes and telling it what to do.
The hypothalamus pituitary gonadal axis, or HPG axis for short. This is a classic endocrine feedback loop.
Okay, so we have gonadotropin-releasing hormone, GnRH, from the hypothalamus, which travels down to the anterior pituitary and stimulates the release of LH and FSH, luteinizing hormone and follicle-stimulating hormone.
Yeah.
But there's a crucial nuance here about how GnRH is delivered. It's not a steady stream, is it?
No, and this is absolutely critical for the system to function. GnRH is not secreted as a constant drip. It's secreted in pulses, a little burst every 1 to 3 hours.
Why pulses? Why not just leave the tap on and provide a steady signal?
Because of a fundamental phenomenon in endocrinology called receptor downregulation, if you were to flood the pituitary cells with a constant high level of GnRH, the cells would essentially become deaf to the signal. They desensitize. The receptors on their surface would literally be pulled inside the cell and degraded.
So too much signal paradoxically leads to no signal at all.
Exactly. You need the pulse on, off to keep the system responsive. It's like a flashing light gets your attention much better than a steady one that you just get used to.
And that pulsatility is key for some clinical treatments too, right?
Absolutely. If we want to shut down this axis, say for prostate cancer treatment, we can give a long-acting GnRH agonist. It floods the system, causes that downregulation, and effectively performs a chemical castration.
Okay, so the GnRH pulses, which means the LH and FSH pulse in response.
LH, in particular, follows the GnRH pulses very closely. FSH is a bit of a smoother, slower response.
And this is where your dual-track control system concept comes in, because LH and FSH have very different jobs when they get down to the testes.
Right. It's helpful to think of them as two parallel but interconnected control loops. Track 1 is the LH-Leydig cell testosterone loop.
So LH targets the Leydig cells, our loud neighbors.
Yes. LH is the signal that tells the loud neighbors to start shouting. It hits its G-protein-coupled receptor on the Leydig cell surface, drives up intracellular CAMP, which activates protein kinase A, and that whole cascade stimulates the enzymes that convert cholesterol into testosterone.
Simple enough. Make the hormone.
Make the hormone. And that testosterone then enters the bloodstream and travels back up to the brain, where it exerts negative feedback on both the hypothalamus, reducing GnRH pulses, and on the pituitary itself, making it less sensitive to GnRH.
So that's track 1. A classic thermostat loop. If testosterone gets too high, it tells the brain to turn down the heat.
Precisely. Now, track 2 is the FSH-Sertoli cell loop. FSH targets the Sertoli cells, our nurses, inside the tubule. It doesn't directly stimulate testosterone production. It tells the Sertoli cells, get the factory ready, support spermatogenesis.
And this is where it gets really clever, I think. The Sertoli cells, in response to FSH, produce something called ABP, androgen-binding protein.
This is the absolute linchpin of the whole operation. This is the key that makes fertility possible.
Explain this. Why do we need a special protein just to hold on to testosterone? It's already right there in the testes.
Okay. Testosterone is a lipid. It's a steroid hormone. That means it can diffuse through cell membranes incredibly easily. It doesn't stay put. If the Leydig cells just produced testosterone into the interstitial fluid, most of it would just diffuse out into the blood and be washed away into the systemic circulation.
Right. It would dilute into the entire 5-liter volume of blood in the body.
But here's the problem. Spermatogenesis, the actual process of making sperm, requires a concentration of testosterone inside the tubule that is astronomically high.
Yeah.
We're talking about levels that are maybe 100 times higher than what's circulating in your blood.
100 times.
At least. If you had that level of testosterone systemically in your whole body, you'd be toxic. You'd have massive side effects. You need that high concentration to be strictly local.
So you need a way to trap it right where it's needed.
Exactly. And that's ABP's job. The Sertoli cells make ABP under the influence of FSH and secrete it into the fluid of the seminiferous tubule. The ABP sits there and acts like a molecular sponge. It grabs the testosterone as it diffuses in from the Leydig cells and it holds it there, creating the super concentrated androgen bath right where the developing sperm are.
It's a testosterone trap.
It is. Without ADP, you simply cannot maintain the high local androgen concentration needed for fertility, even if your systemic blood testosterone is perfectly normal.
Wow. That's a critical distinction. So you could have a guy with big muscles and a deep voice, all the systemic signs of normal testosterone. But if his Sertoli cells aren't making enough ADP, he could still be infertile.
Theoretically, yes. Local concentration is king. It's not about the level in the arm. It's about the level inside that tubule.
And does this second track, the FSH track, have its own feedback loop?
It does. But since Sertoli cells don't make testosterone, they can't use that for feedback. Instead, as they get stimulated by FSH and support sperm production, they produce a different hormone called inhibin, specifically inhibin B in males.
Inhibin travels up to the pituitary.
And it selectively inhibits FSH secretion. It's the Sertoli cell's way of telling the pituitary, okay, we've got the message. The factory is running well. Sperm production is up to speed. You can ease off the FSH signal now.
So this creates a situation where you can diagnose problems just with blood work.
It's incredibly useful. For example, if a man has primary testicular damage, say from an infection or chemotherapy, and his sperm production plummets, the Sertoli cells get damaged and stop making inhibin B.
So inhibin B levels in the blood will be low.
And with that negative feedback gone, the pituitary starts screaming FSH. So you'll see a blood test with a very high FSH level. But if the Leydig cells are okay, his LH and testosterone might be completely normal.
So clinically, an isolated high FSH points you directly to a problem with the factory floor, with the Sertoli cells and spermatogenesis itself.
Exactly. It helps you localize the lesion just by looking at the hormones. It allows you to peer inside the testes without ever having to do a biopsy. It's elegant.
Let's transition from the control room to the hormone itself. Section 4, the molecule of man, testosterone.
The big one.
We know it's derived from cholesterol.
Like all steroid hormones. But as Ganon points out, the testes lack the specific enzymes like 11 and 21 hydroxylase that you find in the adrenal cortex. So the biochemical pathway is shunted away from making cortisol or aldosterone and specifically towards making androgens.
Yeah. Once it's made and gets into the blood, it doesn't just float around freely, right? You mentioned dilution.
No, the vast majority is bound to carrier proteins, about 98% of it. Roughly 65% is tightly bound to a specific protein called gonadal steroid-binding globulin, or GBG, and about 33% is loosely bound to albumin.
Which means only about 2% is actually free and biologically active at any given moment?
Correct. The bound testosterone acts as a large reservoir. The free fraction is what can actually diffuse into cells and exert its effects, which is why, clinically, measuring free T is often a more accurate reflection of a person's androgen status than just total T.
Now, here is where things get really, really interesting for me. I, like many people, always thought testosterone just did everything. But the sources make a huge deal about its metabolite, dihydrotestosterone, or DHT.
Yes, this is a critical concept. In many ways, you can think of testosterone as a prohormone. It's the raw material. In many of its key target tissues, like the prostate gland, the skin and hair follicles, and the external genitalia during development, testosterone is converted into DHT.
And this conversion is done by an enzyme called 5-alpha reductase.
Exactly. And DHT is the heavy hitter. It's the amplifier. DHT binds to the same androgen receptor as testosterone, but it does so with much higher affinity, and it forms a more stable complex. It's a significantly more potent androgenic signal.
And the distinction between what testosterone does versus what DHT does is vital for understanding male development and also for pharmacology.
It's absolutely crucial. Broadly speaking, testosterone itself is responsible for the internal plumbing during fetal development. It maintains the Wolffian ducts, which become the vas deferens and epididymis. It's also responsible for muscle mass, bone density, libido, and the deepening of the voice at puberty.
Okay, so T is internal structures and systemic anabolic effects.
Right. DHT, on the other hand, is the master of the external. It's responsible for the differentiation of the external genitalia, the penis and scrotum in the fetus. Later in life, it drives prostate growth, contributes to acne, and is the primary culprit in male pattern baldness.
And the sources mention a specific, incredible clinical case that proves this distinction beyond any doubt.
The gamete.
The penis at 12 syndrome. It's a famous series of case studies from a remote village in the Dominican Republic.
Tell us about this. It's a wild story.
So in this population, there's a relatively high incidence of a rare genetic condition, a deficiency in the 5-alpha reductase enzyme. These individuals are genetically male, XY. They have tests, and those tests produce testosterone.
But they can't convert that testosterone to DHT.
Right. So what happens in the womb? They have testosterone, so their internal Wolffian ducts develop normally. But they lack DHT, so their external genitalia don't masculinize. They're born with ambiguous or female-appearing genitalia. They're often raised as girls.
Until puberty. Until puberty. Then the HPG axis wakes up with a vengeance. The hypothalamus and pituitary start pumping out LH, and the testes respond by producing massive amounts of testosterone.
The loud neighbors wake up and start really shouting.
Exactly. And that pubertal surge of testosterone is so powerful that in many tissues it can overcome the need for DHT. The clitoris-like phallus grows into a small penis. The testes descend, the voice deepens, muscles develop. These children who are raised as girls virilize and transform into young men almost overnight.
Gavedoses, which translates to eggs at 12.
Exactly. It's a dramatic and powerful proof of concept. It proves the distinct roles of T versus DHT in development.
And it also explains how certain drugs work.
Precisely. Drugs like finasteride or dutasteride are 5-alpha reductase inhibitors. If you want to stop hair loss or shrink an enlarged prostate, both DHT-driven processes, you can block that enzyme. You reduce DHT levels while mostly preserving the systemic levels of testosterone.
So you can target the unwanted effects while keeping the desired ones, like muscle mass and libido.
That's the goal, though biology is rarely that clean-cut, and side effects can and do occur because the systems are all interconnected.
One last little surprise in this section for me. Estrogen. We're conditioned to think of it as the female hormone. But Genong says that in men, 80% of our estradiol comes from converting testosterone peripherally.
Yes. Men actually have quite a lot of estrogen circulating. We make it in our fat cells, in our liver, in our brains, by using an enzyme called aromatase to convert testosterone into estradiol. But here's the kicker. We absolutely need it for fertility.
We do. How?
Well, the testes produce a lot of fluid along with the sperm. It needs to be reabsorbed to concentrate the sperm before they enter the epididymis. That fluid reabsorption mechanism in the efferent ductules is driven by estrogen acting on estrogen receptors.
So no estrogen, no fluid reabsorption.
Exactly. If you create a mouse model where you knock out the aromatase enzyme or the estrogen receptor, the male mice are infertile. Their sperm are fine, but they're incredibly diluted because the fluid can't be reabsorbed properly.
So a man needs a little bit of the female hormone to be fully male and fertile.
Physiology doesn't care about our cultural gender labels. It just uses the molecules that work for the job at hand, and estrogen is a critical, essential player in male reproductive function.
Okay, moving on to section 5, the mechanics of delivery. Or, as I started thinking of it, the semen cocktail.
The semen cocktail is a good way to put it.
Because it's really not just sperm. In fact, sperm makes up a tiny fraction of the total volume, maybe 10% or less.
Correct. The rest of the fluid, the seminal plasma, is life support, fuel, and logistics. It's all about ensuring the sperm survive the journey and reach their destination.
So break down the recipe for us. Who are the main contributors?
Okay. First, and the biggest contributor by volume, you have the seminal vesicles. They provide about 60% of the total volume, and their contribution is rich in three key things. First, fructose.
Sugar. Fuel for the journey.
High-octane fuel for the mitochondria in the sperm tail. Second, they provide prostaglandins. These are signaling molecules that are thought to interact with the female reproductive tract, maybe making the cervical mucus more receptive, and even causing reverse peristalsis in the uterus to help pull the sperm inward.
And the third thing?
Fibrinogen, which is a clotting protein, the same precursor we have in our blood.
Okay, hold that thought. Next up is the prostate gland.
Right. The prostate contributes about 30% of the volume, and its fluid is crucial. First, it's alkaline.
It's a base, so it's there to neutralize acid.
Yes. The residual fluid in the male urethra and the vas deferens can be acidic, and the vaginal environment is definitely acidic. Sperm hate acid, they become immotile and die in it. The alkaline prostatic fluid acts as a buffer and protects them.
And what else does the prostate add to the cocktail?
It adds a clotting enzyme, and paradoxically, a declotting enzyme. The declotting enzyme is prostate-specific antigen, or PSA.
Okay, wait. The seminal vesicles add a clotting factor, and the prostate adds both a clotting enzyme and a declotting enzyme. That sounds completely contradictory. Why are we clotting and declotting at the same time?
It's all about timing. Think about the physics of the situation. Immediately upon ejaculation, you want the semen to stay put. You want it to adhere to the cervix and not just immediately run out due to gravity. So the fibrinogen from the seminal vesicles and the clotting enzyme from the prostate work together to form a coagulum, a temporary gel.
It anchors the payload near the target.
Exactly. But if it stayed a gel forever, the sperm would be trapped. They're stuck in jello. They can't swim out. So about 15 to 30 minutes later, the PSA enzyme gets to work and breaks down the clot. This process is called liquefaction.
And once the gel liquefies, the sperm are released and can begin their journey.
And off they go. It's a beautifully designed, timed release mechanism. Clot first, then release.
That is brilliant. I genuinely never knew that's how it worked.
It's why, when we do a fertility workup in the lab, a standard part of the semen analysis is noting the time to liquefaction. If it doesn't liquefy properly, that can be a cause of infertility.
Now, to get this cocktail delivered, we need the mechanics of erection and ejaculation, the classic point-and-shoot mechanism.
A mnemonic that has gotten countless med students through their exams. Point is parasympathetic. Shoot is sympathetic.
So erection is the parasympathetic point.
Right. And it's fundamentally a vascular event. It's about blood flow. Triggers, which can be psychic from the brain or tactile from physical stimulation, cause parasympathetic nerves to release their neurotransmitters. The key one here is nitric oxide, NO.
We hear a lot about nitric oxide in the context of cardiovascular health and supplements.
It's a potent, short-acting vasodilator. It diffuses into the smooth muscle cells in the walls of the arteries of the penis and activates an enzyme called guanylyl cyclase,
Which then makes a second messenger, CGMP.
Exactly. And the rise in CGMP causes the smooth muscle to relax. The arteries open up wide, blood rushes into the spongy erectile tissues, and as those tissues expand, they press on and compress the veins that normally drain the blood.
So you trap the blood. Easy in, hard out.
That's the mechanism. It's a hydraulic system.
And this is exactly where drugs like Viagra come in.
Precisely. The erection goes away when an enzyme called phosphodiesterase 5, or PDE5, breaks down the CGMP. Sildenafil, which is Viagra, is a PDE5 inhibitor. It blocks that enzyme.
So CGMP levels stay high, the smooth muscle stays relaxed, and the erection is sustained.
Correct. It doesn't create an erection out of thin air. It requires initial stimulation to get the NO and CGMP going. But once it's there, Viagra helps keep it there.
Ganong had a funny little side note about one of the side effects of Viagra.
The blue vision, cyanopsia.
Yeah. Why does that happen?
Well, the target is PDE5. But the drug isn't perfectly specific. There's a very similar enzyme in the photoreceptor cells of the retina called PDE6. Sometimes the drug spills over and inhibits PDE6 as well, which can lead to a temporary harmless alteration in color vision, often a blue-green tint.
So everything looks a bit blue for a while?
A small price to pay for the intended effect, perhaps, for some people.
And then you have ejaculation, the sympathetic shoot.
Right. This is a spinal cord reflex primarily mediated by sympathetic nerves from the T12 to L2 level of the spinal cord.
And it happens in two distinct parts, right? Emission and ejaculation proper.
Yes. Emission is the first stage. It's the contraction of the smooth muscle in the vas deferens, the prostate, and the seminal vesicles to dump all the components of the semen cocktail into the internal urethra and mix them together. That's the point of no return.
That's the sensation of inevitability that precedes the event.
Correct. Then, ejaculation proper is a somatic reflex. It's the spinal cord triggering rhythmic contractions of the skeletal muscles at the base of the penis, specifically the bulbocavernosus muscle, to powerfully propel the fluid out.
And one final crucial step in the life of a sperm, capacitation.
Which, very surprisingly, doesn't happen in the male body at all.
So the sperm leaves the male still not fully ready to fertilize.
Not quite. It can move, but it can't fertilize. It still has that acrosome helmet we talked about, but that helmet is reinforced with cholesterol, making it very stable and tough. It's too hard to break open. The safety is still on.
So what happens? Where does the final activation occur?
In the female reproductive tract. The fluids in the uterus and fallopian tubes wash away inhibitory factors from the seminal plasma and leach some of the cholesterol out of the sperm's membrane. This makes the acrosome membrane fragile and ready to rupture.
And it also changes the tail movement.
It does. Calcium rushes into the sperm, which triggers a change in motility from a steady forward swimming to a more frantic, powerful whiplash motion called hyperactivation. Now, and only now, is it a fully capacitated sperm. It's a guided missile with the safety off, ready to penetrate the egg.
It's amazing that the male system has to trust the female system to pull the pin on the grenade, so to speak.
It's a biological partnership. Fertility is rarely a solo act. It's a coordinated dance between two systems.
Okay, let's head into our final section, Section 6, clinical correlates and broader systems.
Where the rubber meets the road?
We touched on this earlier, but it's worth revisiting why it's so hard to make a male contraceptive pill.
It really boils down to the numbers game. In the female system, hormonal contraception has to stop one event ovulation once a month. In the male system, you're trying to stop the production of millions of cells every single day without disrupting the systemic testosterone that's needed for everything else.
It's the difference between stopping a single sniper versus stopping a continuous zombie horde.
That's a vivid but surprisingly accurate analogy. And the blood testis barrier complicates things further. If you use a drug, it has to be able to cross that barrier to have an effect. And if you do something surgical, like a vasectomy, you breach the barrier.
And Ganong mentioned that about 50% of men who have a vasectomy develop anti-sperm antibodies.
They do. Because once you cut the barrier, some sperm can leak into the surrounding tissue. The immune system sees them for the very first time and says, "Whoa, what is this foreign protein?" and mounts an immune response. It usually doesn't cause any systemic health issues, but it can make vasectomy reversal more difficult because even if you reconnect the tubes perfectly, the man's own immune system might still be attacking his sperm.
Then there's the prostate, a common trouble spot for so many men as they age.
Right. BPH, benign prostatic hyperplasia, or an enlarged prostate. We know now this is primarily driven by the accumulation of DHT over a lifetime. That's why 5-alpha reductase inhibitors are a mainstay of treatment. Prostate cancer, similarly, is often androgen dependent, at least initially.
Which is why one of the treatments is androgen deprivation therapy. You starve the tumor of its fuel.
Exactly. You can do that surgically or chemically, using those GnRH agonists we mentioned to shut down the whole HPG axis. You shut down the Leydig cells, testosterone plummets, and the tumor often shrinks.
Now, Guyton threw in a bit of a curveball that I wasn't expecting about the pineal gland.
The third eye?
Yeah. What on earth is the connection between the pineal gland and the brain and the testes?
In humans, the connection is subtle and maybe even vestigial. But in many seasonal breeding animals, like hamsters or sheep, it's absolutely vital. The pineal gland secretes melatonin, and it does so in response to darkness.
So long winter nights mean more melatonin.
Right. And in these animals, high levels of melatonin act on the hypothalamus to inhibit GnRH. It effectively shuts down the reproductive axis during the winter so that they don't give birth in the middle of a snowstorm.
So the testes is ultimately being controlled by the length of the day.
By the light cycle, yes. As Guyton suggests, this seems to be less important in humans who are not seasonal breeders, but the wiring is still there. We know that tumors of the pineal gland can cause either precocious puberty or delayed puberty, so the link, however subtle, still exists.
Finally, let's talk about aging, the so-called male climacteric.
It's a term that's used, but it's very distinct from female menopause. Menopause is a cliff. The ovaries run out of follicles, and hormone production largely ceases over a relatively short period. The male climacteric is a very long, slow slide.
Testosterone levels gradually decline, but they don't drop to zero.
Right. And sperm production can, and often does, continue into a man's 80s or even 90s. So it's not about a cessation of function. It's about a gradual reduction in efficiency. But what's becoming clearer in recent years is that low testosterone in older men is very tightly linked to metabolic syndrome, to obesity, insulin resistance, and type 2 diabetes. It's not just about sex and libido, it's about whole body metabolic health.
It's all connected.
It always is.
So we have covered a lot of ground today. From the 900 coiled tubules and the countercurrent coolers, all the way to the cholesterol-hardened sperm helmet and the timed-release cocktail.
It's a lot to take in. Let's try and synthesize this down.
What are the big take-home messages for you from our deep dive today?
For me, I think it's the multiple layers of protection. The body understands innately that this production line is precious and it's vulnerable. So it builds a temperature-controlled room, the scrotum. It builds a chemical and immunological fortress, the blood testis barrier. And it staffs it with a dedicated squad of nurses, the Sertoli cells, just to keep the process safe.
And the chemical coordination is just beautiful.
That ABP trap, the idea that you have to trap a hormone locally to create a super concentrated environment to make the system work, that one concept explains so much about why systemic hormone levels don't always tell the whole story about fertility.
And for me, the distinction between T and DHT.
Yes. The difference between being male on the inside and appearing male on the outside. It's a fundamental split in function driven by a single enzyme.
My final thought, the thing I'm going to be mulling over later, is that concept we started with. Persistence as an achievement.
Go on.
You look at a man, and physiologically, he just seems stable, constant. But we've just spent an hour unpacking the furious, high-energy industrial process that's required to maintain that stability. The pumps are running, the GnRH pulses are firing, the countercurrent heat exchangers are working nonstop, the cells are dividing millions of times a day.
It takes a phenomenal amount of energy and regulation to look static from the outside.
Exactly. It's like the duck paddling furiously underwater while looking calm on the surface.
Yeah.
In a world of biological rhythms and cycles, the male reproductive system stands out because it has to fight so hard every single second just to stay the same.
That is a beautiful way to put it. Persistence is an achievement, not an accident.
And if you want to see just how complex that fight really is, we really encourage you to go check out the diagrams in Ganong's chapter on the function of the male reproductive system, or in Guyton and Hall's chapter on the topic. The biochemical pathway of testosterone synthesis alone is worth a look.
Absolutely. The visuals really tie it all together.
Thanks for listening to this deep dive. Stay curious.
See you next time.